And we wanted to address the question, how do filth flies contribute to the spread of antimicrobial resistant bacteria? We know, for example, that livestock farming is one of the main producers of antimicrobial resistance, and everybody knows that flies are all over farms, and we thought that from there they might spread and transmit antimicrobial resistant bacteria to humans.
So what did the funding from JPIAMR mean to you in this research project?
Well, the funding was very precious for us because it was a great opportunity for us to get together, to network and to bring the different experts together, because usually they do not work together because they are from different fields. Therefore, it was a very valuable funding. We started in our region in Germany, in Münsterland, and we found that a lot of flies were colonized with antimicrobial resistant bacteria, and we are now doing a study in Nigeria to repeat the same, to figure out what is the burden in low and middle income countries, and to have an idea how relevant is the colonization of flies with antimicrobial resistant bacteria. So we put together in our group people from industrialized countries, from low and middle income countries, and all other areas that are involved in flies, like veterinarians, medical microbiologists, infectious diseases specialists, and entomologists, of course.
Tell us about the routes you were exploring, how flies potentially could carry resistant bacteria to, for example, humans.
Yeah, that is an interesting thing, because flies can transmit bacteria through different transmission routes. For example, the most important route is regurgitation. They ingest bacteria from manure, for example, and these bacteria can then multiply in some parts of the gut of the flies, and they are then regurgitated in numerous cells. And these bacterial cells can then even multiply in the regurgitation spots. So there is they call it bio-enhanced transmission. But there are also other transmission routes possible, like the translocation from the exoskeleton, for example the legs, or through defecation, because the feces of flies still contain a reasonable number of bacteria, also resistant bacteria.
We can all imagine how potential flies who are actually in various places could transmit resistant bacteria. But what are the key findings?
Well, the key findings are that indeed flies can carry antimicrobial resistant bacteria, particularly in the farm environment. And this happens everywhere in the world. For example, in Germany we found several studies reporting the colonization with antimicrobial resistant bacteria, particularly the carbapenemase producing bacteria and the colistin resistant bacteria. And the same was true in a farm in China, where they found exactly the same.
But can you prove that resistant bacteria can be transmitted in the volume, so to say, that we're infected as humans?
Well, it is likely that flies transmit these bacteria to humans and animals as well, but there is no clear study under standardized conditions that prove that flies transmit these bacteria to others. But there are different hints. For example, several studies showed that bacteria in flies share the same genetic background as bacteria from animals and humans. And therefore, it suggests that there is a transmission, at least between bacteria, humans and animals, but the direction of transmission is not clear, because these are only cross-sectional studies without a longitudinal, timely aspect. At the moment, it seems that flies can play a role in the spread of antimicrobial resistant bacteria, but there are still several question marks that need to be addressed to finally answer the question if flies really spread antimicrobial resistant bacteria to humans.
So the next question comes naturally. How will you proceed from this point with the insight you have? What does the future hold for your research?
Well, the main basis how to proceed is to get funds for future research. And we defined several future research questions. And one, for example, is to define the doses that are necessary to colonize and eventually infect humans and animals. And this only can be done in models, in human models and animal models. And the second main research question is to define so-called quantitative microbial risk assessment models to mathematically model the risk of transmission from flies to animals and humans.